Imagine, just for a second, a world where we completely eliminate those endless, agonizing months of manual medical chart reviews. Where you don't have to just sit there staring at a blank screen, agonizing over what clinical question to ask next. What if the medical data itself could actually autonomously generate and test its own hypotheses? Welcome to This Explainer. Today we're diving into an absolutely fascinating concept called the closed loop. We're going to look at what happens when you take two distinct clinical pipelines, a forward one and a backward one, and wire them together to create this semi-automated engine. It's basically a system designed to massively accelerate the scientific method, churning out hypothesis after hypothesis, but, and this is key, without ever sacrificing human oversight. All right, let's just quickly run through our roadmap for today. First, we'll look at the two pipelines, one loop. Then, wiring the system together. Third, the AI guardrails. Fourth, a live clinical example. And finally, the value of new knowledge. Let's jump right into part one, two pipelines, one loop, the anatomy of a machine. So we really need to understand the basic anatomy of this machine before we turn the key. It all rests on two existing one-directional pipelines. Think of them kind of like two different conveyor belts. First, you've got the forward pipeline. This one takes a plain language question from a clinician, resolves it into a coded FHIR cohort, which is just a standardized group of patient records, and spits out the analytics. Basically, it turns a phrase into a specific group of patients. On the flip side, you have the backward pipeline. This one works in total reverse. It starts with a clinical outcome, like, say, a hospital readmission or an ICU escalation, and literally walks a cohort's coded timeline backward to find out what exactly correlates with that specific outcome. Which brings us to part two, wiring the system together, the blueprint of the loop. So what actually happens when we wire these two conveyor belts together? The core innovation here is honestly remarkably elegant. We take the highest ranked output of that backward pipeline, the absolute top finding, and we feed it directly into the input slot of the forward pipeline. And then we just put the whole thing on repeat. Let's see how this builds across 5 specific cyclical phases. Step 1. The backward pass scans dataset A for outcome correlates. Step 2, an orchestrating AI spots a pattern and drafts a query. Step 3, a human steps in to verify it. Step 4, we take that approved query and run a forward pass on a totally different dataset, dataset B. And finally, step 5, human outcome validation. Now, if you map those steps out visually, you're going to see a pretty stark contrast. The machine is handling the heavy computational lifting across 4 fully automated phases. But nestled right in the middle, and then again at the very end, are 2 high-contrast red bars. These are our 2 human checkpoints, and they are absolutely non-negotiable. They're there to ensure strict oversight, clinical plausibility, and to make sure the loop doesn't just, run away with itself. And that naturally leads us into section 3, the AI guardrails, red bars in the machine. Look, there's a lot of totally understandable nervousness around AI in healthcare right now, especially when we talk about generative AI. So we really need to explicitly define the strict boundaries placed on the orchestrating large language model inside this machine. Here is exactly what the LLM does. It operates purely in the seams between our 2 deterministic engines. It reads the ranked lift tables produced by the backward pass. It chooses a finding that clears a preset statistical threshold. It phrases that finding into the next plain language query, and it decides when to stop iterating, maybe because of diminishing returns or just hitting a budget of rounds. That's it. That is its entire job description. But here is what it never does. This is the absolute crucial point, so pay attention. The AI never hallucinates medical data. It never computes a lift ratio. It never decides a cohort matches a concept. It never writes an outcome rate, and it never, ever fabricates a comorbidity link. Every single metric, every lift, rate, cohort count is a direct deterministic read off the analytics layer. Basically, the math is the math. The AI is merely the translator drafting the next question. All right, let's move from the theoretical blueprint to section 4, a live clinical example, the data journey. Let's look at a real-world day in the life of a clinical hypothesis traveling through this exact loop. We'll use a pneumonia cohort for this. So, setting the scene, we initiate a backward pass on a pneumonia cohort in dataset A. Our burning question? We want to know what is driving ICU escalations. The system autonomously goes to work, scanning across all coded lanes, comorbidities, symptoms, interventions, hunting for what correlates most strongly with a patient ending up in the ICU. And here's where the backward pass really flexes its muscles. The system ranks all those findings and flags a massive 2.4 times escalation lift for pneumonia patients who also happen to have chronic kidney disease. Next, the orchestrating LLM takes that top finding and drafts a simple, plain language query. Pneumonia patients with chronic kidney disease. Boom. We now hit our very first human checkpoint. A real human reviewer reads this, sees the cohort size is solid, confirms it's clinically plausible, and hits approve. Now we cross over into the forward pass. The system takes that human-approved phrase and resolves it deterministically against a completely separate dataset, dataset B. Think of it like a totally different slice of a smart health IT sandbox. The analytics run, and the human reviewer checks the math at our final checkpoint. On the original dataset A, the escalation rate was 61%. On the completely independent dataset B, it's 58. The pattern holds. The human validates it. Folks, that is rigorous science happening in a tiny fraction of the time. Which brings us to our final section, the value of new knowledge. Throughput, not truth. We need to be super precise about the actual philosophical and practical value of this system. This loop is not inventing some magical new type of science. Instead, it's massively accelerating the existing scientific method. It's really a game of throughput, not a brand new category of truth. What actually comes out the other end is what we call a replicated correlational lead. That is a well-formed, cross-dataset replicated, human-approved lead that serves as the perfect input to a clinical studies hypothesis section. The incredible value here is for the human researcher. We are literally collapsing months of manual chart review drudgery into two simple, fast checkpoints. You get to start with a pre-filtered, replicated candidate, instead of just staring blankly at an empty page. But we've got to be completely honest about its limitations too. This loop cannot magically produce causation, confounding by indication, where a patient's underlying sickness dictates their treatment. That doesn't just vanish just because we ran a loop twice, right? It also doesn't produce a predictive model. It never fits or calibrates on held-out data. It can't recover missing, uncoded data, like those unlogged hallway conversations between doctors. As they say, absence of evidence is never evidence of absence. And most importantly, it never ever provides a license to skip those two human checkpoints. But here is where the magic truly happens. Once a pattern clears both of those human checkpoints and gets validated, it doesn't just sit in a log gathering dust. It actually loops back. It legitimately re-enters the system as the next seed outcome for a brand new backward pass. The loop literally keeps building on its own prior confirmed findings. What's really fascinating here is how this completely reframes the knowledge we generate. Clinical guidelines are built over years of exhaustive trials. They are our ground truth. A validated loop pattern is simply a second, mechanized path to that exact same destination. It logs stable, citable facts with their exact lifts, datasets, and human sign-offs right alongside traditional textbook knowledge. Which leaves us with a pretty fascinating thought to wrap up on. If we can unleash a closed loop that autonomously builds, tests, and logs its own audit-trailed clinical patterns, constantly layering confirmed facts upon confirmed facts, what will the medical textbooks of tomorrow look like? And maybe more importantly, how much faster will we write them? Thanks for joining me for this explainer.